Today we're going to make a foldable about mitosis. Please feel free to stop and start the video as you need to. So you will need pages 65, 66, and 67 from your packet to complete these notes. You'll also want a red and blue marker or colored pencils. You can use other colors, but you need to pick one color to represent the maternal chromosome. That's the chromosome you inherit from your mother, and one color to represent the paternal chromosomes. That's the chromosome that you inherit from your father. And down below, these are the 3 pages, what they look like. Now, to assemble this giant foldable, cut neatly along the dotted lines, then tape the pages together on the back. Don't tape on the front or it will be hard to write. Okay, so in the cell cycle, there's a stage called interphase where the cell is just growing. And we're going to see the interphase can be broken down into 3 phases or stages, G1, S, and G2. Now, before the S phase, that's what the cell pictured here is looking like, the DNA is really uncondensed and in the form of chromatin. But I'm showing you the chromosomes so we can then compare what happens at the end to what happens at the beginning. Notice that there is only one chromatid per chromosome, since the DNA replication has not yet occurred. And everything is labeled. Here we've got the cell membrane. This whole unit is called a centrosome, and within the centrosome are 2 centrioles, and that's going to help form the spindle fibers. We've got the nucleus. Of course, we've got the nuclear membrane, the nucleolus, and then again we've got 4 chromatids, 2 of them blue and 2 of them red. These 2 blue ones you inherited from your father. These 2 red ones you inherited from your mother. So for example, let's say that this large blue one is chromosome number 1 that you inherited from your father, and this large red one is chromosome number 1 that you inherited from your mother. These smaller ones, this blue one, let's say it's chromosome number 2 and you inherited that from your father, and this small red one is also chromosome 2 that you inherited from your mother. So after the S phase of interphase, let's take a look at what the cell looks like. Again, in reality, the DNA is still uncondensed in the form of chromatin. But there are actually 2 chromatids per chromosome. These sister chromatids should be identical to one another. So by the time you see an actual X, which is what you normally think of when you hear the word chromosome, DNA replication has already occurred. So what I'm telling you is this chromatid is identical in DNA sequence as this chromatid is. And again, this chromatid is identical in DNA sequence as this chromatid. So DNA replication has already occurred. There are also 2 centrosomes. So here's one centrosome and here's another centrosome. And again, within each centrosome are 2 centrioles. And again, that's what's going to form the spindle fibers. Now, there's something called homologous chromosomes that are being shown to you in green. So this is chromosome number 1, and this is chromosome number 1. They contain the same genes, but not necessarily the same version of those genes. So for example, in this top position on chromosome number 1 that you inherited from your father, that might be coding for blood type B. And then over here on chromosome number 1 that you inherited from your mother, that might be coding for blood type A. That might be carrying the allele for A, and over here you're carrying the allele for B. Well, in that case, when we get to the genetics, you'll know that A and B are both dominant, so that's how you get blood type AB. They could also be carrying the same alleles. This could be the allele for B, and this also could be carrying the allele for B. If that's the case, then you would have blood type B. So this large blue one and this large red one, they're both chromosome number 1. They're carrying the same genes, so they're referred to as homologous chromosomes. This small blue one and this small red one, this is chromosome number 2, and this is chromosome number 2. Again, they have the same genes, but they might not have the same versions of genes. but they would also be considered homologous chromosomes. So when we start this stages of mitosis, the first stage of mitosis is prophase. Now the DNA has condensed from the loose chromatin form into packed up chromosome form. Also, the nucleus begins to dissolve, and what that means is the nuclear membrane will start to break down, and the nucleolus will break down. The centrioles are moving further apart to opposite sides of the cell, and you can see that the spindle fibers are also forming. In metaphase of mitosis, I would like for you to think of meta-middle. Here, the chromosomes are lined up at the middle of the cell, or what we call the metaphase plate. So notice how the chromosomes line up in a single file line. And again, you can see that the centrioles have moved to opposite sides of the cell and that the spindle fibers are fully formed. In anaphase of mitosis, I would like for you to think of Anna away, Anna away. The sister chromatids are pulled apart by the spindle fibers and the centrosome. So this sister chromatid has been pulled apart from this sister chromatids by the spindle fibers. So sister chromatids are being pulled to opposite sides of the cell. In telophase mitosis, new nuclei begin to form. So the nuclear membrane will reform in two daughter cells, and the nucleolus reforms. The spindle fibers begin to break down, and something called cytokinesis begins. In other words, in an animal cell, something called a cleavage furrow begins to form. The cell membrane and the cytoplasm begin to pinch apart. This cannot happen in plant cells because they have a rigid cell wall. And so they form something called a cell plate to divide the two cells there. Cytokinesis, there's no picture for this, but it is when two daughter cells pinch completely apart. Cyto means cell, kinesis means to split. Now this is a kind of a pie chart of the entire cell cycle. What you see in green is interphase. So from here all the way to here is interphase. This is when the cell is not dividing, An interphase can be divided into three phases or periods: G1, S, and G2. During the G1 phase of interphase, the cell grows. During the S phase of interphase, DNA replicates. Okay, so this is when DNA replication will occur during the S phase of interphase. During the G2 phase of interphase, the cell will prepare for division. So what's in yellow here is mitosis. Okay, so you have the M phase or mitotic phase, which of course are your stages of mitosis, and then including cytokinesis. So the stages of mitosis include prophase, metaphase, anaphase, and telophase. And then of course cytokinesis is eventually the complete splitting of the cytoplasm to form two daughter cells. So at the end of mitosis, you started off with a parent cell, and at the end of mitosis, you end up with two daughter cells that are identical to each other. And if the parent cell started off as a diploid cell, at the end of mitosis, you end up with two daughter cells that are also diploid. The chromosome number stays the same.